Hello, everyone. I'm Wannakorn Khopanlert from Prince of Songkla University. First of all, I'd like to thank you to TCI for this great opportunity to present the topic entitled Genetic Engineering Technologies Part 2, Delivery Systems. Today's outline includes the overview production of CAR T cells, the gene delivery for T cell engineering, and probably the Q&A session for those students if we have time. So let's begin. This is a picture of the overview production of CAR T cells. You can see that we retrieved the blood from the patients. We isolated just only the T cells, and then we engineer the T cells to express CAR. And after that, we reinfuse back the CAR T cells in order to treat the patients, especially the hematologic malignancies and the novel right now is the solid malignancies. And the one that we are focusing right now is the part that associated with the delivery of gene encoding the CAR to the T cells, including the mRNA transfection, viral vectors, or the transposons that make the T cells express the genes of interest and become the engineered CAR T cells. So basically, we all know that the naked DNA is easily degraded from the system by phagocytosis or the enzymes like nucleases. So that's why we need the vehicle or the carrier, what we call the vector, in order to transfer the transgenes of interest into the target cells. And in the context of gene therapy, gene delivery is classified into viral-based strategies and the non-viral-based strategies. And the viral-based strategy used the parts of the viral blueprint, such as the herpes simplex virus, retrovirus, lentivirus, or adeno-associated virus. And the other one is the non-viral-based strategy that we use depending on the physical or the chemical methods. So this is another summary of the gene delivery systems that it's easier to understand and see from this flowchart. So I will dig deep into each one of it in detail. Regarding the viral-based strategies, especially in T cell engineering, in this table show the US FDA-approved CAR T cells using either lentiviral or retroviral vectors. So the question is, what are these viral vectors and how do they work? And what are the differences between these two vectors? So first of all, I'd like to introduce you guys to the viral replication cycle. You can see this is a retrovirus. The RNA virus binds to the cellular receptors, fuse to the membrane, and release the RNA into the cytoplasm. The RNA genomes are then reverse transcribed into the double-strand DNA, integrated to the host genome, transcribed, translates, and then the accumulation is budded and released from the cells. And the mechanism of the viral genes delivery mimics the cycles, as I mentioned, and to eventually produce the protein of the interest here. So as I mentioned, what are differences between the gamma retrovirus and the lentivirus? This part of the genes delivery is differences as the gamma retrovirus are encapsulated by a highly stable capsid core that it's require the breakdown of the nuclear membrane during the mitosis for integration, while the lentivirus can directly transport into the nucleus. So the lentivirus can integrate it both dividing and non-dividing cells is compared to gamma retrovirus. So can virus cause the infections? Some of you may be curious in these questions. And do you guys think it can cause, yes? If the answer is yes, please raise your hand. If the answer is no, please raise your hand too. Yeah, and the answer is no. Concerning to the viral vector, scientists use only certain parts of the virus, and they do not cause any infections or allow the virus to replicate itself within the body. So I will dig deep more in those viruses that I mentioned. So the first one is the gamma retrovirus. gamma retrovirus is a simple viruses that in the retroviridis family. The most common is the MMRV or the myelonemurine leukemia virus. This is a structure of virus that I mentioned. It's very complicated here, but I try to explain in an easy way. The structure of virus contains 3 major coding segments, including GAG, PO, and envelope, which encode important proteins in viral integration, replication, and encapsulation. And moreover, the pi packaging signal over here is a cyst, exiting RNA element for packaging during the assembly. And this is essential for the production of the infectious particles. And when it's come to gamma retroviral vector, as you guys know, it must be the replication defective by removing the trans-acting elements, as I mentioned, from the genome and replacing by the transgenes of interest, and leaving only the long terminal repeats and the packaging signals. So the vector replication can only occur in the packaging cell lines that still contain the GAG, PON, and envelope, allowing for retroviral to proliferate. And in order to increase the safety by using the viral vector, you can see here, the GAG, PO genes are contained in one plasmid, while the envelope gene is contained in another plasmid. And the prior system for the gamma retrovirus vector using the HEC293 cell lines. However, later on, Gary Nolan from Stanford University, he developed what we call it the Phoenix. It is a second generation packaging cell lines that contain GAG, PON, and envelope in the same one, but safe. And the viral production, it's faster, way faster in just a few days if compared to the own one. However, in the early trial of using the gamma retroviruses vectors, especially against the X-linked SCID, they reviewed about genotoxicity and the development of the leukemia, which is not good, right? So that's why they try to overcome the limitation by explore another vectors, what we call the lentiviruses in gene therapy. So next is the lentivirus. Lentivirus is another retrovirus, but complex. And the most studied is the HIV-1. Again, the genome still contains the same genes as the simple viruses, including the GAG, PO, and the envelope. However, it comprises of more genes, like comprises of more 6 other genes, the 2 regulatory genes, and the 4 accessory genes. So this is the structure of lentiviral vectors. You can see on the left-hand side is the 2nd generation. On the right-hand side is 3rd generation. The way that they try to develop 3rd generation is for the safer, like the safer designs features for the generation of the lentiviral vectors. As you can see here, this is the tables that compare between 2nd and 3rd generation. And you can see that 3rd generation containing more plasmid as the packaging plasmid over here, instead of 1, divided into 2 plasmid. And overall, the plasmid that they use in the system is 4 plasmids. And they delete the regulatory genes, what we call the TAT gene, in the 3rd generations, which TAT gene is responsible for the natural HIV replications. And in the 3rd generation of the lentiviral vectors, always containing what we call the SIN, or the self-inactivating expression vector, that reducing the, in order to have the SIN, it's reduced the proviral mediated gene activation. So overall, the 3rd generations claim to be safer than the 2nd generation. However, it can be more difficult to use because of the lower viral titers due to the additional plasmid, too many plasmid in the 3rd generation. So to generate the CAR T cell using the viral vectors, genes were transduced transfected into the packaging cells, collected for the viral particles, and then transduced into the T cells. And this table also summarizes the viral vectors between those two that I mentioned. And importantly, both of the viral vectors offer stable gene insertion. However, the retroviral, as I mentioned earlier, that in the early trial, that it's associated with the genotoxicity because they study later that retroviral can be integrated in the CPG island, and it causes increasing the risk of the insertional mutagenesis and long-term oncogenesis if compared to lentiviral, that it's more integrated away from the TSS and are considered safer. However, in CAR T cell therapy, so you guys might wonder, is it safe to use both? Because the US FDA already approved both of the lentiviral and the retroviral CAR T cells in order to produce the CAR T cells in the US FDA approved. In terms of the CAR T cell therapy, so far, both are safe to use in the clinical trials. So since there is some concern for the viral-based technology, researchers try to find suitable vectors based on non-viral systems and providing sustainable gene expression without triggering unwanted results. So this table shows the T-cell engineering clinical trials in various cancer here, and using the nucleofiction or electroporation as the delivery methods with the cargo of the transposon DNA plasmid or the mRNA carrier. And the first one is the transposon systems. So the DNA transposon or the transposons are the units of the DNA that can change their position within the genome via a mechanism that we call cut and paste. And it's flanked with, by the inverted terminal repeats. And the trans process, which is the enzymes, will recognize this part of the ITRs and binds to this binding sites. And after that, it's induced the double strand break through the formation of the synaptic complex. The original site, it's rapidly repaired by the host non-homologous enjoining, and the transposon recognizes target DNA and complete the integration. So the important features for the transposon system, it's very simple manufacturing process and very low cost production if compared to the viral vector, because it's involved just only the manipulation of the plasmids and no difficult QC if compared to the viral vectors, as we know. And it's give the permanent genomic insertion leading to sustained and efficient transgene expression as well. However, they reported the lower transfection efficiency if compared to the viral vectors. So the most widely used of the transposon systems are the Sleeping Beauty and the Piggyback. Very beautiful name. So this is the table compare the Sleeping Beauty and the Piggyback. And you can see that the Sleeping Beauty is reconstructed from the inactive transposon sequences in fish genome, while the Piggyback, it's from the cabbage looper moth. You can see that Piggyback show the higher, larger cargo capacity and higher transposition were observed in the Piggyback if compared to Sleeping Beauty. And however, the Piggyback also associated with increased risk of the dysregulation due to its integration profile. And you can see in this table as well that it's extensively optimization to increase the efficiency, including the parental plasmid, the transposon, and the vector delivery methods. And now both of the Sleeping Beauty and the Piggyback are used in the clinical trials. Another one is the mRNA that allowing the expression of the transgenes about 1 week, so it's transient. The mRNA is translated into protein and lost after like 2 to 4 cell divisions, which is why this technique is used in non-proliferating cells. And no concerning of the genotoxicity. Its transient features results in reduce of target effect with lower toxicity. However, poor persistence occur. So that's why in the mRNA-based CAR T cells, they offer the repeat doses were applied for the stability with possible complications that occur with the repeated dose. So in this table also show the clinical trials of the transients of the mRNA CAR T cells. You can see, interestingly, this is from BT and colleagues. They are the the one that doing a lot of mRNA transduced CAR T cells that targeting the mesothelin and found that repetitive infusion transiently persisted and helped with the anti-tumor efficacy. However, severe anaphylactic shock was reported, and they found that it probably due to the murine origin of the scFv. So nowadays, we have the new technology using the nano carrier is also developed. And this is the example of a non-integrating DNA nanovector generating CAR T cells that are active both in vitro and in vivo. And this technology is capable of replicating in the nucleus of the dividing cells. So it leads to persistence of transgenes expression, absence of the genotoxicity, and it's easy, simple, and affordable to produce. And you can see this is some results of the nano technology vectors. You can see that the nano vectors show the enhanced anti-tumor activity compared with the previous integrating vectors, which is the viral vector one here, and prolong overall survival in the mice model. And in this paper, they also show the provide manufacturing protocol in order to quickly generate the CAR T cells with the nano carrier. And right now they are on the clinical trials as well. Finally, it's a gene editing mediated transgene insertion. They use the, the gene editing right now brought the promise of specific and efficient insertion of the large transgenes into the target cells. And there are trials that using the TALENT or the CRISPR-Cas9 in order to insert the transgenes into the CAR T cells. And the details of the gene editing will be explained in the next session by Dr. Chanan. This is some of the examples of the research that they use the CRISPR-Cas9. Very interesting one that they use the CRISPR-Cas9 in order to insert it, the CAR genes and use it in the T cell engineering. They inserted the anti-CD19 CAR at the start of the exon 1 of the T cell receptor alpha chain gene or TRAC. And this TRAC CAR enhanced the tumor rejection with less tonic signaling, less exhaustion marker, with uniform core expressions and greater cytotoxicity against BALL in the murine model. And the other one they used in the solid tumor regression as well, in the solid tumor model as well, that they used the CRISPR-Cas9 in order to insert the anti-GD2 CAR in the TRAC locus. And this anti-GD2 CAR show better cytotoxicity both in vitro and in vivo against the solid tumor. And it's also demonstrated the persistence and less exhaustion related if compared to the retroviral CAR T cells against the solid tumor, which is the neuroblastoma xenograft model. This is the summary of our methods. And the aforementioned, I also mentioned about the transfer method, including the transfection, transduction, and electroporation. And I will explain a little bit more for those students who are may be curious what is the difference between the transfection, transduction, or the electroporation. So basically, transfection and transduction, it's how we introduce the foreign DNA in order to incorporate it into the host cells. The difference between the transfection and transduction is the transfection on the left-hand side deliberately introduce the naked or the purified nucleic acids into the eukaryotic cells, into the membrane cells by using the chemical or the mechanical electroporations or gene guns or microinjection. And on the right-hand side is the transduction. It's a process that deliberately introduces genetic material into another cells by using the viral vectors. And for the electroporations, we use the pulsed high voltage electrical currents to transiently create this small pore over here in order to introduce the mRNA or the plasmid DNA and enter the cells. For the nucleofiction, it's similar to the method of the electroporation, but it's more advanced in order to directly inject it inside the cell, which increase the efficiency of the process and allow the genes transfer to the cell, which probably those kind of genes comparatively hard to transfect. Yeah, and those are the about the delivery systems, and hope you guys enjoy.